So, hello everyone, I'm Pilar Castellanos from Veterans Lab, and today I will be discussing the project A Functional Study of the PLG3, a fifth transposable element domesticated protein in Drosophila melanogaster. Transposable elements are selfish uh, genetic sequences that move and amplify using the encoded proteins of the host and their machinery. Most of the these insertions can be deleterious, sometimes they can be neutral, and in very rare cases they can be beneficial for the host. In the lab, we have been studying these beneficial cases through an evolutionary process called molecular domestication, when the T's protein coding regions are co-opted by the host, acquiring new cellular functions. The Pith-Harpin superfamily of Ts has been domesticated across multiple Drosophila lineages, and right now I'm investigating DPLG3, one of the four DPLGs in Drosophila melanogaster, derived from a Pith element. DPLG3 is located on the third chromosome, is intronless, and is a conserved gene. According to Flybase, DPLG3 is very high expressed in ovaries, but is also expressed in testes and in the nervous system. The Pith-Harbin family has two distinctive open read differences. The first one is the transposase, the second one is the MADEV, and the DPLG3 is derived from the first one. To study the function of the gene, previous studies in the lab used the CRISPR-Cas9 technology to knock out the fly's DPLG3 gene, and then this gene was replaced with a DS red protein in the eye. Also, doing dissections of testes and ovaries, we observe an abnormal or rudimentary phenotype, and we divide in three different categories. The first one are normal ovaries, normal testes. The second one asymmetric, only one is smaller than normal one, and the third one is totally rudimentary or abnormal. These have a higher magnification, but in reality, these ovaries and testes are very small. So this led us to study the PLG3KO in detail and elucidates its role. In figure number 5, we can see a cross made between female DPLG3 homozygotes and male wild types. So that shows the frequency of the progeny of abnormal phenotype in females and males. And we also use the Fisher test to observe if the results were statistically significant, and for both cases were significant. So we conclude that the PLG3 phenotype is recessive, is always observed when the mother is the PLG3KO homozygote, not when the father is homozygote. This phenotype can be completely rudimentary or asymmetrical for both sexes, and is also partially penetrant because we observe it in around 10% of the females and in a small fraction in the males. Also, with the use of HA tag for immunostaining, we observe the protein localization of the PLG3 during oogenesis. So the first panel shows that the PLG3 HA protein is broadly in the germarium and in the somatic cells. The second panel shows that is in the nucleus of the somatic cells, and the third one shows that is in the nucleus of the nerve cells and also in the nucleus of the oocyte. Panel D is a closer view from this one. We can see the negative control. For panel F and H, so we can see the localization in the cytoplasm of the germline. So we also perform RNA-seq analysis for DPLG3KO normal ovaries in the lab, and we analyze the differential gene expression. That analysis showed that 86 genes are upregulated and 37 genes are downregulated. 26 of these genes are involved in digestive plus testes, 14 are testis-specific genes, 20 are nervous system, and also the flamenco locus is upregulated 4 times. So we think that the rudimentary phenotype in DPLG3K ovaries could be explained by the differential expression of male-specific genes that they are not typically expressed in females. So our conclusions and future experiments, we believe that because the PLG3 localizes in the nucleus and still conserves its by DNA domain, it might be acting as a transcription regulator. We also infer that the PLG is important for oogenesis. We believe that the non-rudimentary DPLG3KO gonads are close to become rudimentary because of the expression of some male-specific genes. If that's the case, the misexpression of any of the male-specific genes should occur very early in the gonad development, and we will test this by doing RT-PCR for the early stages in young DPLG3KO females. Right now, we are trying to rescue the abnormal phenotype, but also we are trying to rescue the differential express genes and the T expression changes as well in the DPLG3 KO normal ovaries using a recombinant DPLG3 HA line. We also want to rescue using a very precise approach that is Galford system only with DPLG3 gene. We also plan to produce knockdowns also with GAL4 in the germline, also in the pole cells to see if we observe the KO rudimentary phenotype. Thank you.